we'll see. All right, it started, so let's see if it continues. All right. I don't know why the problem with the transmission. So I do want to finish up the eye. There isn't a lot left. I'm not going crazy with the physiology of it. I'll do a little bit of the other senses, not the ear. I'll save that for next time. We'll dissect an eye. I didn't have a whole lot. I only have about 4 or 5 eye specimens. For those who, and again, dissections, or if you want to just look at it, that's fine. If you don't want to look at it, it's fine too. But you should all observe it, and I'll go over what's involved and give us something to do. That'll be fun to do here. And then next time, I think next week it will involve some kind of. I have some very interesting technology that we use. I'll show you. Again, for your group, it's more for demonstration purposes. It's called iWorks, and it's this thing. So we have an iWorks teaching assistant. And effectively what we can do is we can, and this is what we do a lot in this, measure respiratory, blood pressure, pulse, electrocardiography with sensors like this. So we have a lot, it's all non-invasive. It's nothing that's going to zap anybody or hurt anybody. But it's kind of neat to see how it works technologically, that kind of thing. So I have quite a few, I have some things, and the first one, because it involves the eye and the ear, is a pretty easy one to do, and you can just kind of see. But it takes a little doing on my part to set it up. This is not a great room for it, and I'm going to see whether we can move, at least temporarily, to one of the classrooms if they're unoccupied to do that. And these chairs, just stools, are horrible. So I don't love those either. But hey, what are you going to do? So to that end, if you're feeling uncomfortable and you want to stand up and move around, fine. You get a phone call or something you have to take, go outside. One of the students already came up. She might have to leave a little bit early. We'll be done by that time, however. But if that's the case, just let me know. You can either flip me an email, or you can come up before and just say, hey, I've got an appointment, or whatever. Low key, low maintenance, preferably. All right. So let me, and I did send you something the other, didn't I though? Yes, I did. Oh yeah, what goes on in the lab? It's here somewhere. Oh, this is where that's the other guys that were here. I sent you something with a lab schedule, didn't I? Like the grading thing, correct? Is that ringing a bell? Is there a recollection of that? Perhaps not. Okay. Well, I would have thought that I had sent that, but that's fine. Nevertheless, so this is not there. Should have had that hooked up before. I apologize. All right. Well, good enough. So effectively, what we do in here from a grading perspective, it's different. Really, there's usually 2 practical vehicles. In other words, a practical is basically, and I'm showing you some of the illustrations of labeling exercise, for lack of a better way to describe it. So because we have a relatively full class, I put out about 20 stations with usually 3 or 4 responses. And effectively, we work our way around kind of in a serpentine fashion, kind of like musical chairs, without the music. Unless you want the music, which I'm willing to do. And basically, you have, I'll usually allot a minute or a minute and a half to try to answer this. There's plenty of time afterwards. It's not like you're limited to a minute. Oh my goodness. Because then I don't want you to have the time constraints or anything like that. So it's basically labeling, like the parts of the eye, what we have looked at already. Those, and we have typically one in the middle of the term, one in the final. There are generally 2 quizzes that we do here. They're multiple choice. So I think, and I'll have to look at the layout, and I will send it to you, what the point values are. And then like today, we do a dissection, and we have probably at least 3, I would think that we'll do, maybe 4. Then I'd like a paragraph. It doesn't have to be a whole typewritten. I'd like it typed. You can hard copy it. You can email it. Whatever you want to do. To me, take a couple of weeks to get it done. You're probably busy. As long as I have it in my hot little hands by the end of the term, I'm happy. Okay? See, it's not hard. I have other things to do. Just saying. I'm not big. So it's like 2 quizzes, 2 practicals, seeing your bright smiling, or at least your face is here. It's always good from an academic perspective. If someone comes to you with a problem 6 weeks, 8 weeks into the term, oh my God, I'm not doing well. It's nice if you recognize their face. It's when you don't that it's a problem. They haven't been there. I'm sure you've heard that before. We call it Stein's theory of academic success. Just show up and sit there. Osmosis. Nevertheless, here we go. So and I was recording all that nonsense. Clicky. So that's the introductory material. This is kind of where we left off. I do want to finish the eye and a few little odds and ends here. They're fairly straightforward because smell and taste, there's not a whole lot going on, okay, that are there for those special senses comparatively, because it's really just chemicals. I should that. Interesting. Color blindness, less likely and much less likely in women than in men because it's sex linked. If you're familiar with your genetics, sex linked, it typically means it's passed along on the X chromosome. So therefore, and women with 2 X chromosomes, basically we have effectively it masks. Typically, it's unusual to find 2 recessive characteristics of this type. It could happen. It's just relatively speaking uncommon. They give it you, a fairly large percentage of males, and it's varying degrees. It's the one with, you've seen it with like the little dots that you look at, the colored dots, typically red and green, and you can't see, like it would be a number 7, for instance, or a 57. It's typically the one that you see. And there's a particular name of those. We have them somewhere in here, like that. My dad had it. Okay, but again, it's on my mom's side, they didn't have it. So that's, genetically speaking, I did not have that. So the most common type is the red-green, where either one or the other are absent. And so it's a little bit different. It's not extraordinarily problematic, depending on what your occupation is. But it's just a neat little aside. Genetic things are interesting. Okay, there's a lot of very interesting genetic disorders that surprise you that they're more common than you think, or at least variations of it. This is the key item. This is the vitamin A aspect of things. It's called retinal, and there are 4 different proteins, 3 associated with the color receptors, 1 with the rods, effectively the black and white receptor. So, and they're classically referred to as opsins. So they are rhodopsin, and the R tells you it's the rod. And then there's other ones, different kind of opsins that are based on the 3 colors. So since there's a certain amount of overlap, you can get more than 1 color. And that really is sort of that amazing richness we have with our color vision, to be able to discern all this array of different colors. It's just fascinating, to me. And then there's different forms, but the key is right here, cis and trans. Cis is bent, trans is straight, okay? So when it's dark, it's bent, but when it absorbs light, whatever the light is, it straightens out, and that's the whole ballgame, in a nutshell, of you being able to see something, okay? Conversion of bent to straight initiates reaction that leads to electrical impulses. And it's what I was saying before. Normally, that circuit is working, and when it's working, it doesn't allow the optic nerve to fire. It's sort of working in reverse. It's what we call counterintuitive. Again, normally you think when I see something, it's going to trigger a response. Here, what it does, the response is it blocks a nerve circuit pathway, but that's a sort of a repressive pathway that then allows you to visualize. So it's rather than depolarizing, it's hyperpolarizing scientifically. So by making that pathway no longer working the way it was, we're able to see something. Okay. So it's called phototransduction, light basically changing shape, creating energy. Okay. So the rhodopsin, the pigments are deep purple, and it goes through a series of steps. We make it, we undergo a process called bleaching and then regeneration. And it's very similar in the cones, but it's a little bit easier to understand in the rods. So in the dark, you make more of this. When you're asleep, you make more of this. When it absorbs the light, they call it bleaching, and it goes from the bent rest form in the dark to the straight form. Okay? And as well as triggering that electrical response, such as it is, or hyperpolarizing that electrical response, they separate or break down. And then eventually it's all converted back to the cis form again to start the whole process again. So we're constantly, if you want to think of it, recycling a lot of these elements that are there. And they show it to you in some degree of detail, none of which you have to know the chemical formula. You don't have to do this type of thing. You don't want to do that. I was a chemistry major. This is kind of fun for me. I don't even remember it. It's not fun for you. I know. It's okay. The chemistry majors are a strange bunch. We don't even have them here. So we just have chemistry. We don't have majors in chemistry. They're not wired the same way as the rest of us. So as you can see here, it has this and it goes straight. So it's really a subtle change if you want to look at it globally. And it goes through a particular process and they give you all the steps and you don't have to know it. So it activates. I don't know whether, did they talk to you about these G protein pathways? Yes or no? Is that ringing a bell at all? Okay. There's two ways that we signal things in the human body. Okay. You can signal things from outside a cell or inside a cell. So certain things trigger changes. For instance, if you know somebody who has a severe allergic or an immunological condition, or maybe they're infertile and they have taking steroids, or they're like my son and daughter-in-law went through IVF. Okay, there's a lot of steroids. Okay, those are fat soluble. They get directly into the cell and actually make their way into the nucleus eventually to trigger responses in the form of eventually leading to creation of proteins by way of translation. Okay, so it goes and it really initiates what's called transcription and translations on a DNA RNA basis. Almost everything else, if you know someone's a diabetic, and we all do, it takes insulin. That works through a G pathway. If your body produces human growth hormone, that works through a G pathway. So those are basically proteins. They're too big to enter the cell. So what they do is it ends up going through this kind of a process, which thankfully you don't have to know. Okay, but they'll show it to you. So in the light, it uses an energy molecule. Our energy molecule is ATP. ATP and GTP are very similar. They both have a similar chemical basis, biochemical. One is, you remember the A's, the T's, the G's, and the C's, all that DNA stuff? The A's and the G's are very, very similar. They're called purines. So adenine and guanine behave similarly. And so they're showing this is a classic pathway. Here's the light. The light changes the shape. It goes through a series of different proteins along the interior of the membrane within that rod or cone cell, whatever it is, eventually leading to these kinds of changes. So we begin to see in the dark, the channel is open. And in the light, the channel closes. That's what's meant by hyperpolarization. Okay. Normally that channel is open, and that activation is keeping that particular visual pathway from being active. Once it closes, it activates. It's like a suppressor pathway. But again, I'm not asking you to know all this. I'm just giving it to you for context because it's unusual. So it goes through all, and this is the way So in my advanced physiology class, to the pre-med students, we talk about EPSPs and IPS, which are excitatory and inhibitory postsynaptic potentials, which I'm sure you don't want to know. No, she doesn't want to know that either. Were you here for the cat picture? Curiosity and the cat. That's a cat who's curious. We had a cat picture. You missed the cat picture? Can't have you missing the cat picture. You never can tell what you're going to get from Amazon. So he never met a closed door he could tolerate. Let me out. I want out. Right away. 4 o'clock in the morning, I'm hungry and I want out. 6 o'clock in the morning, I'm hungry, I want back in. And I want back in. So we call him Mr. In and Out. For a silly cat, he was a rescue. My stepdaughter fostered cats. He didn't get along with her cat, who we call Miss Meanie. And so, will you keep the cat until we find a home? Guess what? 2 days later, our cat. Nevertheless, so I don't go into all the EPSPs and IPSTs, but effectively what it does, it all involves neurotransmitter. A classic neurotransmitter, glutamate in this case, is a hyperpolarizing agent, usually works the opposite, and that's how you see. And they show you at length the pathway. In the dark, the channels and the transmitters being released, and you can't activate the pathway. But in the light, it changes and it does activate the pathway. So if you're interested, you can look through that if you want. If you're not, that's fine. We adapt. Bleaching occurs even in a little bit of light. So again, rods are very sensitive to any light. So in bright light, it happens very quickly. So we are able to we're able to adapt. And the same thing with our hearing, the same thing with our smell, and with our taste. We have adaptability. The circuits become less sensitive. It's like you can hear it here in the lab, that terrible noise. That's always there. Eventually you kind of tune that out. It's like a bad smell. Maybe you're in the micro lab, and sometimes it gets a little funky in there with all the cultures. You can tune that out. We all do that. So when moving from darkness to bright light, we get a glare because it's not just the cones that respond, the rods respond even more so. And so it breaks down and your pupils constrict, and it takes a while, over 5 to 10 minutes, depending to some extent genetically, how rapidly you'll respond. Those kinds of things. Years ago in school, we were in dark a lot because we didn't have PowerPoints and things like that. You had film strips and slides, and we used to sit in dark rooms with slides. It was long before the technology caught up, and the technology is a big help. And the same thing, bright to darkness, okay? Other different things occur as well. Your pupils dilate. And again, you'll increase. For most of us, it takes a little while. I take it fairly long. So that I have a certain degree of night blindness. It doesn't affect driving. In me, fortunately, there are others that it does. And so a lot of times vitamin A deficiency. This is a very interesting disease you do not want to have. Retinitis pigmentosa, known as RP. Okay, it's a degenerative disease. The rods are not replaced. A neighbor of ours, of my first set of in-laws, she developed this in her 20s, and rather than have children biologically, I do not know if it was genetically based, but she was an adoptive parent, as am I. Did I tell you all my kids are adopted? Tell you? I actually chose that. What was I thinking? So what I'm saying is, in order to not have that genetic thing follow her family around, because she was completely blind, and he was a kind of person, close neighbor and friend. We celebrate holidays with. She was widowed fairly young, that type of thing. And so this is how it gets there. Now you may remember this, you may not. Okay, depending on how detailed your AP1 was. It probably was done more in the lab than lecture. I don't know. And I'm not being critical of anybody. I'm trying to give you this for context. I'm not going to ask you to tell me that the optic tracts go to the lateral geniculate bodies of the thalamus and then the radiation in the cortex. I'm going to give you the idea of what goes on. Remember when I spoke loudly in class? That was the superior click light. You know, a sudden light or a sudden sound, you turn your head. Superior is light or visual, something stimulus. Sound is the inferior colliculus. And there's a whole lot of these. This is what it all means. And I did this when I did graduate anatomy for nerves. I taught at Chatham for a number of years. You know, we did something like this. These are called visual fields. We test. You get your eyes tested, see if you're missing something, and you can predict based on, not that we're going to have to do it, I'm just giving you again for context. So the outer portion, let's say, of your left eye sees broadly here. The outer portion of your right eye here. Less so, as you can see, you really are quite a ways over to see from the right over to here and the left to here. And as you'll see, most things cross over in the human body. My left side of my brain making my right hand, and vice versa. In your eye, it's about 90%. So about 90% cross over in what's called the optic chiasm. You know what that is across? About 10% do not. And you can even predict what 10% do not. And then those fibers, they all eventually go to an area in your brain that's effectively a relay center that you might remember called the thalamus. It's part of the diencephalon. Okay, and that basically is what it is. It relays sensation to the appropriate area, smell, taste, and in case of vision, this is called the optic tract to the thalamus. From the thalamus, there's a structure called the optic radiation that sends it back here to the occipital lobe where you process it. And then effectively, that's a lot of dedication to processing those images and then associating it with things that you've seen in the past, just the way a child would learn. If it's a cat, if it's a dog, if it's brown, if it's black, whatever. So we can, if there's an area of localized blindness, we can actually predict with this where, and the term that we use is called a lesion, the irregularity, something that's there that should not ought to be there. So here's dissected out. Optic nerve, optic chiasm, optic tract, thalamic area, and then the optic radiation back to the occipital lobe. So there's nothing unusual about the pathway. So needless to say, it's like you're always worried if somebody falls backwards, hits the back of their head, you know, you're more at risk of having some type of visual problem or even transient blindness in the other way. My kids were all skaters, hockey players and figure skaters. My daughter's figure skater, boys were hockey players. And even when they did learn to skate, it was always, well, everybody had a hat on in case they fell, until they kind of got their sea legs, that type of thing. So, and because of the fact that it's called stereoscopic vision, you get input from both eyes, and that's when somebody has lost the side of an eye, has to wear a patch for whatever reason, whatever the situation, they lose a certain amount of depth perception. It doesn't mean they can't drive or can't function. Okay, my brother-in-law lost an eye about 40 years ago from a bungee cord hitting him in the eye. Okay, and still does all that stuff. He's a few years older than I. So I get that's the idea. Okay, so then strokes can sometimes affect it, which are damaging the brain itself from a lack of blood supply. And so and that's all the stuff. That's really everything from this. It gets processed back in the primary visual cortex. All of these things, then we go to what are called association areas that are moving a little more forward, and it's passed on going toward the frontal lobe where effectively we process. If I see, it's like I'm looking out at you, and if I see someone who you might have a question, I'll notice it and respond to it, or I might try to make some comment. We're constantly doing that. Done with the eye, you're happy, ecstatic. Smell and taste are basically the same thing. Chemical receptors. When we're done with that, we'll be done with having to listen to me extensively. Chemical receptors are just that. They have to dissolve. So in the nose, for smell, we have mucus, which has moisture base, and we have saliva in our mouth. So we're basically breaking those chemicals down to some type of ionic component, a charged particle that will trigger a response. So, and a lot of times they work together. So probably know very well, if you have a cold, your nose is all stuffed up, things don't necessarily taste the same. Okay? And maybe if you have something in your mouth that has a coating agent, or maybe something that's irritated, it may impact the way that you smell and taste things. So there are dissolved chemicals, hence chemoreceptors. Same deal. Bipolar cells have to take that chemical signal and create that into some type of electrical signal that goes to somewhere in your brain to tell you what it is. So in your nose, you have respiratory structures, but you have a fair amount of olfactory epithelium, lining cells that are about smell. More in the roof of your mouth, and because if you remember your bone anatomy, there's a lot of little nooks and crannies. They're called conchae. Now, if you take my dog, please, he's a poodle nose. I mean, there's a nose. I mean, he could stick his nose in your pocket from over there. He does. But animals frequently have a much greater sense of smell. They have more surface area. So that's why we can use scent-trained dogs for a variety of things. Okay. So we learned early on that if you're going someplace, be careful in those days what you had in your luggage. These were in the ages before marijuana was somewhat legal. I started college the summer right after Woodstock. That was a rock concert of some renown 54 years ago. Was it ringing? Didn't go. Sold tents for people who did go. So all of that, you have these sensory cells and bipolar neuron. So you start with the olfactory, the sensory smell, just like in the eye. It goes to a bipolar, and then it basically goes in. So you remember, hopefully, there's the ethmoid bone, about the deepest bone in your skull. It has the cristagalli, it's like cross-shaped. So you have the cristagalli, which the meninges are attached to, perpendicular plate, which is the upper part of the nasal septa, the bony nasal septa, and then sort of like this on either side, as you'll see, it has what's called a cribriform plate with little foramina, little openings for these nerves to go down and sort of wend their way into the nasal cavity, which is much bigger than you thought. So they're bipolar in nature. Specialized knobs are really the sensors, covered by mucus, and they're all part of the first cranial or olfactory nerve, and you can get new ones a lot. So if something really affects your sense of smell, something harsh, maybe smoke, maybe particles, typically your sense of smell will come back. Smell is really important, everybody. Smell triggers memories. Smell goes directly into a system that you might have learned about called your limbic system. That triggers emotions. Things that smell good, things that smell bad, remind you of things, either good or bad. Smells are a big, there's almost like an emotional investiture. And that's the limbic system, has very much tied with your sort of emotional well-being. Okay? So here they're going through the cribriform foramina. Here are these more or less bipolar cells, and down here, sort of the olfactory epithelium are sort of specialized little knobs on the end. And so lots of different odorants. This is an old number, Some of the stuff I'm reading suggests that we have several thousand of these, so we have a greater amount than you would think. Okay, and they bind, and then different things, and that's what we use. There's pain and temperature receptors, so we use things. That's what smelling salts are. So if you're going to be in a clinical field, so for some of you start an IV and somebody kind of faints, and they're going to, trust me, lying down or not, they're going to faint. Okay. How do you get them to wake up? Smelling salts. Now, I don't see it today, but in my office, and I had an office for seeing patients for 25 years approximately, we would on the wall have a padded tongue blade in case somebody started to sneeze. Okay. And we'd have a little packet of smelling salts in case somebody coughed. Because that's how your first step to revive them. Normally, if they faint, that's strong cell. So it's crack it, a little ampule inside, spirits of ammonia. You could use different things. So it has to be somewhat volatile and able to dissolve. And then more of these G protein receptors. I'm not going to go into all the subtleties and chemistry of it. And that's basic. Then we have olfactory adaptation. So if you're exposed for a long while, you'll adapt. And we do the same thing with sound and with taste. Same kind of a pathway. Not much else to tell you about that. It goes to a relatively, it's why we use smelling salts to make somebody alert, is part of that nerve goes right in to the frontal lobe of your brain. So, I mean, it's going to be part of your, it's called a reticular activating system. Okay? Some other smells may go through, but here, hypothalamus, amygdala, and other regions of the limbic system, emotional responses to odors. And some people who don't smell can be an early sign of Alzheimer's. Remember in COVID, we saw people losing smell and taste. Some people had demonstrated that when they had that. I'm not quite sure what the derivation was of that. Olfactory hallucinations, okay, are sometimes people, if they have seizure disorders, they'll have a smell and it'll kind of, so watch out for that. If you have a patient who has a history of seizure disorders, it wouldn't hurt to ask them what, because then generally they get what's called an oral. They get a warning. That's a warning so they can, make themselves ready. I mean, it's very frightening. Have you ever seen somebody undergo a grand mal seizure? Now we call them tonic-clonic seizures. Scary. Taste, very similar, very interesting anatomically. We have thousands of them. There's different kinds. I'm not going to ask you to know the difference between fungiform, looks like a mushroom, valate, V-shaped, foliate, etc. And they're all over the place. Cheeks, tongue, palate, not just on the tongue. So they're all over, as you can see there. And what's interesting, they have these kind of interesting lining cells that make for little clefts. And the cells kind of come to a point, and we call them, and you're going to see this again when we start to look at sound, called hair cells. So they're really not hairs, but they kind of look like they narrow, and there's little openings for taste. Okay, different neurotransmitters that are involved. So they're kind of in here. So here you get this sort of V-shaped in this cleft, and then there's little openings where the dissolved substances will trigger a response. They kind of look like this. See these sort of, they call them hairs, but in reality, they're just a lot of surface hair. Classically, the four major: sweet, sour, salt, and bitter. Sweet, typically, and salty are really in the anterior portion of the tongue. Sour, things like I cook. So what do when you want to? You always try to use a combination of all of these. So you use sweet things. If you're making a dressing or a sauce, very famously, there's one called an agrodolce in Italian cuisine, sweet and sour. You use vinegar, you use sugar. Sometimes you'll make a reduction out of it. The French chefs call that a gastrique. Impressing you with my cooking acumen. If I have a third career, I'll be cooking. And then, so sweet, sour, we put salt on almost everything because it's a flavor enhancer. And even sometimes bitter things have that, but you feel like you taste it mostly in medicines and stuff like that. Like a pill starts to dissolve that you take, it's like. But we have a palate for all of those, meaning it's enhanceable. Umami is very interesting. It derives its name from glutamic acid, which is one of the principal amino acids and a neurotransmitter. So, and most famously in Asian cuisine for years, they used a flavor enhancer called monosodium glutamate, known forever as MSG. You could buy it in the grocery store. It came as a powder called Accent, and you'd sprinkle it on stuff. The trouble is that glutamate in certain people would produce, it would be such a stimulant, they'd start to perspire, their face would turn red, etc. Yeah. So now most of the places you go, you kind of stay away from that. If you go to an upscale, right, you could definitely stay away from it. Don't like that stuff? Love that. That's umami. So you get it from certain things that'll have certain kinds of mushrooms will have that. They'll enhance things. Okay, there's an interesting sort of flavor components to add to that. You know, like Thai cuisine fish sauce, Worcestershire sauce, which is made from anchovies, all have that umami. That's what you derive it. Soy sauce. I used last night. Oyster sauce. This is another one. Some people really have a palate for fatty foods, long chain fatty acids. That can be problematic. And even some folks really, they almost need foods to be extremely spicy or hot. We even have sensors for that. And you choose different peppers in cooking because they have what's called a finish. They'll trigger like heat taste buds on your epiglottis. So if you like to cook, and I like to kick it up. Okay. It's like last night. I mean, I can't resist throwing the, like a little chili flake into almost everything. Just saying. That's all. That's all. Fairly quick adaptation. Not a whole lot else to say. We're going to go over this, not today. Next week, I think I'll do the cranial nerves. Your tongue's interesting because you have a facial portion, facial nerve, anterior 2/3, and a throat portion, glossopharyngeal nerve, the ninth cranial nerve. So you have the seventh and the ninth innervatus. This is, again, none of those PowerPoint errors. It should have a little I there. It's 9, not 10. The vagus is the 10. That's one for the epiglottis. Done. Now, let's do the eye and consider it. And I'll go over this with you. I have a handout over there for you to look at. Hopefully this will come, that sheep eye dissection. So it should be on there. I have the link. It comes from it. I have it on the paper if you're interested. So just if you Google sheep eye dissection and go to Burroughs, you'll get this. So what it is the eye is a very interesting anatomical structure in the sense that and it's a really good little 10 or 15 minute dissection. It tells you what you really want to see in relatively short time. So depending on the eyes that you have there, I'll leave this up. Go to the computer. They look kind of like this. They're relatively small. It's easier to have a towel. Okay. So depending on how good these are, I can really get this towel. You see it getting squished. Which is a problem. Anything like that. They put these, it's called carrier wonder. I don't know how dissection. Did you do the brain dissection? They pack these things done called carosate. Carolina Labs, we buy this stuff. This is an agent that's much safer than in the old days. Everything was in formaldehyde. So the carosate, theoretically, you should for form, I throw it away. So I have a couple others that he didn't look really good. They're not individually packed, you can use them. So when you open that up, inevitably, even though it's supposed to be safe, I rinse it. Get a dissection tray and I'll pull off the instruments. And I'll show you as you go from there. So we have four of them. Divide yourself up. I wish I had more to go around. I told you to do it anyway. So at the second thing, I don't need safety goggles. I got more gloves with micro lab. Okay. Now I'll get you the instruments that are out. So you can take a look at it. So you should be able to see. These probably don't have the eyelid apparatus, but you'll be able to get your orientation when you see, start to see, for instance, like this one. It'll be kind of like this. This would be the stump of the optic nerve. This would be more or less the area where the cornea is, the pupil, and the iris are. So kind of when you cut it, we're going to make sort of a cut. I'll show it to you in a second when we move this along. You should be able to see that. Sometimes, and the ones we used to get, they were loosely packed. You actually have little stumps of those extra optic nerves. I'm getting there. So you'll be able to, you may or may not want to remove some of the fat to see this a little bit better. This effectively is the corneal area, okay, that covers it, even though it'll be clouded over. And this is the stump of the optic nerve. So once you get the orientation like that, you kind of want to make a cut, as you can see it this way. So when I say the orient, it's like going from the North Pole to the South Pole, if you want to think of it that way. Okay, and you can use, yeah, I've seen they do it with razor blades. I like scalpels. And so I have sharp scalpels, and if not, I'll replace the blades. I'll replace the blades, not you. Because if you get cut, it's a problem. If I get cut, it's not a big deal. Sorry. I mean, when you open it up, if you do it carefully, when you cut in, it will sometimes you'll get some of the fluid from that vitreous humor will come out. It will not hurt you. Don't be standing over it like that in case it decides to squirt. Okay? The whole squirt thing's bothers you. When you open it up on one side, and I wish it was a little bit better, on one side you'll be able to see that serrated edge, and you'll be able to see the lens probably, at the very least, you'll see the iris and you'll see the pupil. On the other side, you should have that membrane for the retina. You actually can see the fibers heading toward the blind spot. If you carefully sort of lift out, almost like tease out that packet of vitreous humor, underneath it you will see this. We do not have this in our eye. This white area that you can see here and here, that's called the tapetum. It's kind of like a reflector. So if you, like my dog's collar has it. It's like if someone goes out at night and they're jogging and it's twilight, they'll have something reflective on, or guys working and gals working on the roads at night. Animals have that. It amplifies their light. Their vision at night is considerably better than ours. So it's like they have an internal reflector. So you will probably see that as well. Now on the other side, so here you can see the remnants of that ciliary body. On the lens itself, you can see those little pigmented areas are actually suspensory ligaments, the pupil, the iris, and you'll see the cornea even though it'll be clouded over. So all of that stuff will be there. I mean, they do a whole worksheet. I don't care. So I'll hand them out and you can just pass them around. Here they are. So, I mean, there's the website if you're interested in it. So identify the major features of the eye. House externally, stub of the optic nerve, any muscle renders prior to the wire. When you look at it on the inside, you notice the three layers, fibers out, they're called tunics. You see this in blood vessels as well. They use the term tunic, and we still use it occasionally. You see the sclera cornea, the choroid layer, and the retinal layer. And I mean, it's just one page, paragraph. I'd rather have an impression about it. You help me understand the eye, that's part of it. So you can start guessing these around. And over here, I have dissecting trays. We'll need like 4 of those. I'll bring them out. Here. I'll put some blades on these. The ones that I can replace with blades. So you'll see some forceps. You and I would call them tweezers. The official name is forceps. Scissors. You'll see some probes that are there. These blades are not particularly good. I have three or so blade handles that I can use that'll have good blades on it. Very big. And gloves, I have plenty of smalls. I have a new box of mediums I'm just opening now. Don't have a lot of larges. I have a new box of extra large. That's all I can find. Bring over to the lab. They're all, latex free. So. So let me replace some of these blades. Go ahead. Those things are sterile, by the way. I want you to know those. There. Here's the new one. I got 3. So wherever you want to divide yourselves. Again, it's more up to you to do. I just want everybody to be able to see it so we can point out the structure. Ways of working here. We're actually supposed to put these on the way instead. Well, good. So I have 3 new blades on there. We'll have 4 eyes. So if we come up and be courageous and get started, you don't feel like we just look. Throw away all this stuff here. Put these away. Okay, open the package. Use your scissors over there. And then we can rinse it out and go from there. So like I said, so we have 4. We have 1 more eye. For those who are interested, I'm not. Everybody should take a look. Excuse me, my slide by. Strength here. Let me. Hopefully this will work.